So I'm going to talk about measurement assurance for rare event analysis. Okay, this is on. It's been a whole IT day from hell for me. Okay, what's she doing? Okay, sorry about that. So I'm going to start out today talking about some new resources. Then I want to introduce you to a couple of concepts: measurement assurance and design control. And then finally, I will get to how we will apply that to a rare event analysis. So hopefully most of you are aware, we have a new guidance document out last year on the validation of assays conducted by flow cytometry. And I don't have too much time to go over this, but I want to introduce the document to you and let you know why it might be relevant and why it should be a your new Bible in your lab. So it's CLSI is the Clinical and Laboratory Standards Institute. So I've bolded "and" there because it's, while clinically oriented, it's not just for clinical laboratories. It's a nonprofit organization that develops laboratory standards, and they're a global leader in laboratory standards, recognized by regulatory agencies, clinical labs accredited, and it's aligned with ISO. So basically, this document is, the target audience is anyone that touches a flow cytometer. And it covers all aspects of flow testing. So from uncrating your instrument to archiving your results. I'm really going over this part quickly. We have representation from a lot of flow cytometry experts, from ICCS Clinical Cytometry, American Association of Pharmaceutical Scientists, CAP, ESCA, ISAC, and we have a global representation. Part of the CLSI process is you need 2 representatives from each of these constituencies on your document writing committee. So we had FDA and NIST representing the government. Industry would be pharma or instrument and reagent manufacturers we had present. And professional would be those of us that work in the lab, CRO. We had rep from the NIH and various clinical labs. We need a flow cytometry specific document because the existing documents for validation of other technologies are not fully applicable to flow cytometry, primarily because we're working with live cells and the fact that our data aren't derived from a standard curve. I don't really have too much time to go into that today. Why we went with CLSI for the document, first of all, it's because it's a consensus document. It's aligned with ISO, and the regulatory agencies, including the FDA, often recognize the documents or part of the documents, and they were on our writing committee. This is a long slide, but it's just going over the writing and review process for the documents. Okay, there we are. So we formed a committee, which I showed you the representation of, and then the committee, as well as CLSI, vote on the document. Can this draft go forward? And then this is the critical thing. It's open for public comment. We received a ton of comments. And part of the process then is every comment we had to address and send our responses back to the submitter. And then they had the opportunity to appeal our resolutions. And we did get some appeals, including from the FDA, and we went back and forth with them to resolve their questions and make sure that they were fully answered. And so that's why we went with CLSI, and why a consensus document has a little more weight behind it than other recommendation papers. We also did in this document, we didn't include anything that was not already published in a peer-reviewed journal. So you see some of the recommendation papers we've done in the past, they're represented here, although there might be some differences. Okay. Okay, so I'm going to talk in a minute about this concept of measurement assurance, but briefly we will look at the elements of a measurement, right? And consider all of the aspects. So in flow cytometry, it's our instrument, our method, how we report, monitor, and document. And so the CLSI document goes through all of these, it considers all of those aspects. And I also, as resources, want to bring your attention to, this was late last year, a special issue of Clinical Cytometry geared towards a next generation drug development. And this is completely open access. And there's two papers in particular, one on high sensitivity flow validation, which we'll go over today, and also method transfer. And those are good resources aligned with this presentation. Oh, it's been so long since I've had to fiddle with all this stuff. Okay, so measurement assurance and measurement science. This slide is from a NIST presentation that I went to a few years ago. And it's an approach that informs us on the comparability and confidence in our results. And so it enables us to know how much confidence to have in our measurement. And so the concept is to consider the components of the measurement. So it's a value, and that allows us to compare it to something else. There's some uncertainty in the value, which we're going to get from our validation. And then there's evidence that we've got confidence in our system. And so this is what we get from all of our nice quality control processes that we do in the laboratory. Another aspect of this is to think about our assay as a measurement process. So to build measurement assurance, and we actually have a talk on this that's still Googleable, but looking at everything we do in the laboratory, basically, and then building a process around that, and then deciding what are the key indicators for each of those processes. How would we monitor those, and what's our specifications for accepting that each of those processes has passed the criteria. Okay, so again, we're going to look at the measurement assurance. And so you would basically in your laboratory have a process for each of these things, right? Now we're going to introduce the concept of design control really briefly. And this is a process I just really became aware of recently, and I wish I had known about earlier in my career. I think a lot of things I figured out, but it would have been nice to be aware that there was a process for this. So just a framework of practices that's used in regulated environments for assay design, development, and validation. And you would incorporate this into, the first step would be to identify the needs of your customer or your assay, and define the context of use. So I'm going to talk about context of use. I highlighted in red, so we remember that a lot during the presentation. And then the next step is the design input. So define your assay performance requirements. So the process looks like this. I'm going to go through this quickly. So we would assemble a team and define our needs and goals as we start. So now we're thinking about developing rare event assays. And even if you're not in a regulated lab, your research or something, you're never going to do anything on your own. So your team might be, you and the PI or you in the lab, and just thinking about what are we going to do. And it's always good to get input from other sources. Then define the scope and determine the feasibility of your assay, and then the design process. We all know how to design and optimize flow cytometry assays. Validation, read H62, we all know how to validate flow cytometry assays. And then we run our assay. And what's really important in the process is this lessons learned project review. So is there a process improvement opportunity? Okay, so I'm going to pull this process together into designing and validating and implementing rare event assays. All right, So thinking about what do we want our assay to do, and this relates a bit the next few slides, what Sophie was just talking about. Early on, think about what's my final context of use. So your initial considerations is what do I want to measure in what matrix, what patient population if you're doing something, but even in a research, so what biological system. Your additional logistics would be what decisions will my data enable? And what's the life cycle of my assay? Like today, it might be in a drug discovery setting, but later it might move into a clinical trial, or you might be in research setting, but you hope it goes to be a diagnostic test in a clinical lab. So be thinking about that early. And what are your regulatory requirements, if any? There might be none today, but as the assay goes on its life cycle, there might be some. And this is something to think about early when we design our assays. So the next process is method design, development, and optimization. So I'm trying to tie this back to the elements of our measurement. So for rare events, in terms of our instrument, we need to really think about optimization, standardization, and carryover. And then in terms of the method, we want to really optimize our method when we do and develop it properly, and look at, for sure, the precision and the limits of quantitation when we do the validation. There's other things, but these are the things that we'll highlight today as they're important for rare events. Okay, so we don't have that much time today, and this is a talk in of itself. So I've just put some papers there dealing with instrument optimization, and I think most of us know how to do that. Instrument standardization, even if you have a single instrument and you don't have multiple instruments and multiple laboratories that you'll be doing your testing in, you should have a standardized process for how you set up your results every day, how you set your PMTs, and this will give you better consistency from experiment to experiment. But if you've got more than one instrument or more than one lab, you absolutely should be doing this. It will absolutely decrease your variability. I'll show you some examples. So there's a lot of processes for this. Again, it's a talk in of itself. And some of the newer instruments have built-in mechanisms for transferring assays and optimizing. And then there's a new NIST consortium on flow cytometry standards, and we're looking at evaluating some new processes and making use of the NIST ERF beads. So next year at the meeting, we definitely should be inviting Lily Wang to give us an update. We have some big interlaboratory studies that have just started. And these are my favorite papers for instrument standardization. So here's a quick example. We did this when I was at Covance. So we've got 3 Fax Canto 2s. And in the blue color, it's just the standard setup using your CS&T beads. Every instrument's well maintained, it's a regulated lab, and passes the daily CS&T. We then ran some hard dyed beads on each instrument and looked at the MFI. So this is pretty interesting. So you could have up to 50–60% difference in your MFI readout on instruments that are not cross-standardized to each other. We then, BD had developed a method for cross-standardizing these instruments, and we put that in place. So we ran that process and then put the same beads back on, and you can see how this readout, the variability is really decreased. And so this is why it's worthwhile to cross-standardize your instruments. Carryover, again, in thinking of that context of use, we're thinking about today rare events. So yeah, if we're carrying over some events from the last sample, we don't want that. So we need to really look at that in a rare event assay. And we talk about this in H62, so I won't really go into it today. And this slide, when I talk about validation, I always have this kind of slide up, but I don't spend much time on it. But now for rare event assays, we really want to think ahead of time. And again, like what Sophie was saying, when you design your assay, know what you'll be doing. And so we need to really think about, I'm not loving this guy. Okay. Okay, think about how we'll define our population for our rare events. The positive selection, negative selection. We need to really think more deeply now about our fluorophore selection and our monoclonal antibodies clone evaluation. And so as we're designing the assay, how we're going to do the gating, what our outputs will be, this is really going to influence what type of rare event assay we're able to generate. And then we need to fully optimize our panel, which we may not normally do as much, but looking at our lice, fish, fix, wash sequences, and look seriously at how many events we need to collect for the rare event assay. So when you define your population of interest, I'm going to design a new rare event assay. You need to look to the literature. What are the current phenotypes for that particular population? And they change over time. What's the recommended gating strategy? I look to the omic panels a lot from ISAC and to see how people have gated, that they've looked at different clones. Also look at the literature. There's a lot of recommendations. The most common rare event assays are minimal residual disease for leukemia, lymphoma, CD34 stem cells, CAR-T, PNH, circulating tumor, and plasma cell measurements. So go to the literature and see what people are recommending as your starting point. You still need to evaluate things, but you need to be fully aware. And the reason I say that, it may seem like so obvious, but I'm an editor for Cytometry B, and I'll look at some of the papers we get, and people are not as well informed with the literature as they should be, because I'm aware of papers that they haven't cited in their work. Okay, I love this slide. So when I first started doing flow in a regulated lab at Bristol Myers doing biomarkers, I joined the Purdue website, and there was this post from Mario Roeder, huge letters: stop using CD45RA with RO to define naive and memory populations. And man, this scared the life out of me. But the fact is, this deals with, if we think in terms of validation, assay specificity. Okay, am I measuring what I think I'm measuring? And so here, in 1990, CD45RA was defined as identifying naive T cells, and RO as defining memory. Then in 1995, papers came out and said, you know what? The RA positives, there's also effector cells in that population. So if you use that marker alone to define your naive cells, you will not be specific. You're also going to have some effector cells in there. And then also the other markers that you could use to identify this population were defined. And so I guess I have to agree with Mario. I would be using huge letters as well. If 5 years later, people have still not read the literature and incorporated this. And stuff changes quickly, especially nowadays with all these new dyes and new instruments, new buffers that we have. So you really need to do your homework, and even more so with rare event assays. Okay, the gating, just knowing again, are the gates, those events in my gate what I think they are? In the interest of time, I won't go over this too much, but you need to understand your biology so that you've excluded cells which might not be the population you're interested in. So here it's a monocytes, and we've got CD16 and CD14. This gate's upstream is set incorrectly. You're going to pull in some granulocytes, which are CD16 bright. Those aren't monocytes, so we need to exclude those from our gate. Here, if we wanted to measure CD4 T cells from lymphocytes, you would gate on your lymphs, gate on your CD4 and H, and this is very specific. If this gate were set incorrectly and you also had monocytes in there, then you would have some CD4 dim cells. So that's not going to be specific. So you need to understand your biology. This example is from a different CLSI document, and this is a really good document. A performance of single cell immune responses. Phil McCoy and Holden Macker were the document chairs for this. This will be going up for renewal next year. So if you guys are interested in participating in the renewal of that document, keep an eye out on my LinkedIn. I'll let people know that you could join the committee if you wanted. It's a really good learning experience. So here the point they make in the document. So we consider intracellular cytokine staining can be a rare event. And they point out the importance of setting your gates correctly and knowing your biology. So CD4 is downregulated in activation. The activated cells are producing gamma interferon. So we need to include the CD4 dim cells in our gate. And here they have not. And you can see we're getting different results. And so this gating is not specific enough. We're losing some of our sensitivity of the assay. We're not getting all of our cells. This is an example of a rare event assay we did at Covance. It's in the public domain already, but we wanted to measure B cells after a B cell depleting therapy in a clinical trial. So normally B cells would not be considered rare events, but here we were going to look for B cell depletion, and the difference between 0.5% and 2% would not be clinically significant, but in the purpose of the trial, that's going to matter. And so we needed to develop high sensitivity, so an assay for rare event B cells. So we included 3 B cell markers and lots of dump channels. We also included an isotype control, which normally you would not need for B cells, right? They're a distinct lineage. You can find them with CD19 alone and no FMO or FMX control. But in a depleted situation, we wanted to be sure that we were consistently setting our gates. And so we did include I call it an FMX, so control. So what I'm going to show you here is a nice example as you develop your assays. So we gated, this is not the gating we used in the assay. I just actually regated for the purpose of a presentation. We got CD4 lymphs, and then we deselect the first dump channel, the second dump channel, and then we have some few contaminating cells, and we're able to gate on the CD19 positive. Now, if we did not do this and just gated on lymphs and CD19, so really the only markers you need here, you can see here in my CD19 positive population, I've got some of these cells and some of these cells as well. Now, thinking of our context of use, if I am in normal B cell ranges, is that level of contamination meaningful? Not at all. If I am in depleted B cells and I really need to know how many, did we knock out all the B cells, this level of contamination will impact my assay. So these are things to think about. Clone evaluation, we all know how important that is. And even in any situation, I can't imagine picking this clone if I had evaluated this clone here. And this slide is courtesy of Anaga Divekar at BioLegend, as is this one. This one I like because sometimes when people do clone evaluations, they'll just do single color. And here we see the impact of looking at CD38 versus IgD. I'm not seeing anything that tells me which clone will be better. Now, if I gate CD27, which was in the assay, this is the plots we would have used, versus CD38, you can see here, this is the characteristic clustering that you see for the transitional B cells. They're not popping out here. And this, you can imagine, trying to set this gate consistently is going to be difficult. And so obviously you're going to pick that clone. So again, when we evaluate our clones, we need to put them in the right context. Fluorophores. I don't know, is anyone else just obsessed with all the new dyes coming out? I'm constantly trying to read about them, and I want to try them. And this I got off the BD website recently. Per CP versus CD279, those are the results. And with one of their new dyes, BB700, I mean, this is astounding. I don't know why we would not use that dye in our system. So again, even more important in a rare event setting to do our homework. Now, method optimization. I don't know, I don't do this enough, I will confess, but in this example, we wanted to look at CAR T cells. So for development, we're able to take normal blood and spike in CAR T. We know there will be situations where the CAR T is going to be highly represented and situations where we need to monitor where there are going to be rare events. And so we need to carefully develop our assay. So it doesn't matter what these protocols were, but these were actually from the literature, what clinical labs often use. And one of them was the good old pre-lyse and then stain. And you can see here, we've got 1% background and here 1.3% essentially, which is not bad. I mean, that's okay. But here we can go down to 0% background. And this is the lysis type procedure we want to bring forward. And I'm going to show you when we validate limits of detection. I'm going to circle back to these plots. Okay, these data are from Christoph Eberl, one of my new colleagues at Charles River. And he has compared, this is 3 different assays. I'm not going to explain the assays to you because it doesn't matter. The point is, he has compared centrifugation with laminar wash, and a vendor today is Curio. I have no financial disclosures to share about Curio, but I think it's really cool, the data, and I wanted to share it. And also I have to give a plug for the ISAC Cyto Innovation Committee, which I used to chair, because this technology was first shown in the technology showcase at Cyto. I forget which one. But here you can see the differences in washing the cells, the traditional centrifugation, or with the laminar flow. And this example is, to me, like, wow. So depending on what your system is, you may want to evaluate this type of cell washing in your assay. And you can go to the booth, and I am going to give them a little plug and talk to them about that. But I really thought these results were extraordinary. Now, this slide has been around forever. It's from 2010. So they, Mike Keeney and someone else came out with this example looking at how many events do you want to collect for your rare event assay. So they did some math, and your desired CV is 1% up to 40. And my population of interest is 10% down to very, very small. And then they calculated, to get those CVs, how many cells should you collect in your assay. This is a great guide when you design your assay and you've developed it, and now you want to know where should my stop gate be on what population. This is not a substitute for validating the CV of the assay. And we gave a workshop, I think it was Cyto Boston, on rare events, and somebody like yelled out at me that, you know, the workshop was useless because this paper was already out. And it's like, no, we're talking about something different when we talk about validating the assay. Not that I carry a grudge. Okay, so now let's switch into validation. Okay, so these are the typical validation parameters: accuracy or trueness, specificity, sensitivity, precision, QC ranges, linearity, stability, carryover, and reference intervals. So we already talked about our assay, what the dots in our gate are. And that's really established during assay development. You can't do more in validation, but you need to describe it. And that's our specificity of the assay. All of those good practices that we do in flow cytometry. You could look for interfering substances during validation. Sensitivity, that validation terms change, and the new term is detection capability. I'm not responsible for the terminology, but I just want to share it with you so you know. But everyone still says sensitivity. So this is influenced, as you saw, by our assay development and optimization, how sensitive our assay will be. But we can demonstrate that during validation, and precision. So not forgetting that the definition of validation is that we're going to demonstrate through experimental proof that the assay meets its needs, its context of use, or just even characterizing what is the assay performance. So that's what we're going to do when we validate. There's different flavors of precision: intra-assay, repeatability is a new term, inter-assay, reproducibility. We can look at between analysts or between instruments. And we would just look at our results and establish what the variation is, the CV. So if we look at intra-assay precision, and this is, it's the most important, and I'll show you why in a minute, but it's really easy to do. We would just get 3 samples, and we're going to assay in triplicate and do everything in triplicate: stain, acquire, gate, and in the same run, and we calculate the CVs. And people always wonder, what is an acceptable CV? And it depends, again, on our context of use, as well as the cell type. So this is our total B cells here. And you can see we've nicely clustered from the negative CD19 negative population, and there's a good number of events there. So you will achieve 5 to 10% CV with this kind of data, these kind of data. Now here for plasma cells, okay, there's not very many, and it's not such a clean population. Like, it's defined as 27 bright and 38 bright. So do I gate here? Do I gate there? What about these guys? There's going to be more variability in that population. And we just want to characterize it and understand what that variability is. So what intra-assay precision tells us is this is as good as it gets, right? My plasma cells are going to have 35% CV. My B cells are going to have 5. Now, when I want to look at stability, different reagents, different instruments, different analysts, that's my yardstick that I can compare things to. And I can tell you, I've worked with some academic labs, and they'll just pull a number out of a hat, like, okay, should be 10%. And it's like, no. We need to characterize it. And you can have different, so we're getting the plasma cells and the B cells out of the same tube, the same assay. We can accept different CVs for each reportable result. Okay, now let's get back to rare events. I set a timer here. Let's see if I can figure out how much time I have left. Okay, we're good. In a regular validation, people often overlook assay sensitivity or detection capability. They don't do this in the validation, but it's really important. We need to know at what point do our values get imprecise. And to validate that, there's 3 terms we consider: limit of blank, limit of detection, and limit of quantification. So the limit of blank, so it's harder to conceptualize for flow cytometry, but if you're doing an immunoassay and you have PBS instead of your cell supernatant or your plasma, that would be your blank, right? You have everything in there but your analyte. And you just calculate that, the mean, and I didn't invent these numbers, but the standard way is 1.65 standard deviations. And then to do the limit of detection, you can just determine that empirically, or you can just calculate it and use the mean of your blank plus 3SD. So that's pretty easy. And then we need to figure out, when we have our cells there, at what point do they become imprecise? And that's our limit of quantification. And like I said, this is optional if you're not going to be in rare events, but it's mandatory if you're doing a rare event assay. And I'll show you some examples of how applying LLOQ can improve your data set as well as your interlaboratory comparability. So how would you do LOB and LOD in flow cytometry? So and you have to think about this. And then again, this is why it's nice to have that initial discussion and thinking things out and have a team, because you're never going to think of everything. This example is easy. If we had a true FMO, that would be like our buffer blank, right? So here we have B cells, and we want to look at activation with CD69. So we have a real FMO here. There's no CD69 APC here. And you can see, oh, probably not, but there's some events there. So we're going to have some background. And this would be our blank for that type of assay. And then another way you can do it if you're doing an MRD assay for leukemia or for something you're going to spike in, like the CAR T cell example I showed you, then your unspiked cells. So for leukemia, you could always use a normal. And for the CAR, this is really easier to understand. We had spiked earlier CAR T cells into whole blood. So these are the two different lysis procedures I showed you. And you can see here, I'm going to take the mean of 10 experiments and add 3 SD for my limit of detection. It's going to be pretty high here if you've got 1.26% background versus here when you've got 0. So this is what we want to establish for our assay. Now the LLOQ for stain cells, this is a little bit harder. And the hardest thing is figuring out what samples we're going to use and how we're going to do that. If, like with the infused cell products, that's easy, right? We can take whole blood and spike in various levels of CAR T cell, or for MRD, we can take normal and spike in leukemia samples. And I'll show you examples for that B cell assay I showed you, and it's just one population. So we're able to deplete it with immunomagnetic beads and admix back in. So that was easy. But if we're looking at a phenotyping assay, then it becomes harder, or more extensive phenotyping, a subsetting assay. So here what I usually do is I'll fully stain one sample, wash out the unbound, and partially stain another, and then kind of admix them together. And so it just takes a little bit of thought. Running the samples is pretty easy. You would just do, I like 1 and 3 dilutions. This is not written in stone, but I think that gives me a nice spread. 5 levels, 3 donors. Now, when we analyze the data, unfortunately, there's no graph or simple calculation we can do to understand what our limit of detection is. And so what we look at is we need to look at I don't like this pointer. Okay, we need to look at the population clustering as well as the numeric results. And we're going to look at the number of events as well as our reported result, like my percent of plasma cells. And with these dilutions, we need to make sure that there's a dilution, a titration effect, because if you're no longer seeing that, then you're below your limit of quantification. So let me show you an example of this. So here's a graph that we cannot tell everything from the percent positive versus the CV. This is a cytokine, intracellular cytokine on CD4 T cells. And in this example, PBMCs resting were spiked with activated. It's only 2 donors, 6 levels, and 1 to 2 dilutions. So I wouldn't have designed it this way, but it's still very useful. We have to make sure that all the dilutions from donor 1 are assayed on the same day and donor 2 on the same day. We then calculate our LOD, and here we decided to threshold at 20 events. There is no magic number here. It's what you want to do and what your rationale is and what you can demonstrate as acceptable. Some labs randomly use 100 events. I picked 20 because from my experience, I can see good clustering and get reliable data when I have only 20 events. And I, in some cases, could have gone lower, I think, but I was just not comfortable doing that. And just take the high road and say, okay, I'm going to set it at 20 events where I know I've got confidence in the data I'm producing. So the same data we showed you there, this is just for one of the cytokines. What we did here is donor 1, donor 2, each of the levels. And this is the number of events. and then the frequency of cytokine positive cells. So first thing we do is we highlight all the values that are below 20 events. I'm not going to consider those in the analysis. We calculated our LOD, which I can't remember what it was, and I highlighted those because these are below the LLOD. Now, your LLOQ can be the same as your LOD, but it cannot be lower. So then we looked at our replicates, the mean value, and the CVs. So for this donor, I got 22% CV at 0.27% positive, and for this donor, 10.3% CV and 0.029% positive. So those values were very close. We've got good CVs, and if you do 3 donors, it's better. You can have a little bit more confidence, but this was certainly acceptable. So that was, we set the limit of quantification at 0.27. Okay, we're still in our design control process, and we're in the home stretch. Going for my assay's approved, I'm ready to run it, and I'm going to give you an example of that, and then the self-assessment lessons learned. So you're reporting your assay, so you have to figure out what your reporting limits are going to be and how your data will be reported when you're below your limit of quantification. Also, you need to understand, and this is true, I'm kind of clinical trial oriented, and I apologize, but even if you're doing research as well, you would still be thinking of these things. It's like, well, will I consider those data in my biological system that I'm investigating if they don't meet these criteria? And you need to establish how you'll report your data when your minimum number of required events are not met. And in clinical laboratories especially, is we need to understand the context of use, like what data are going to be enabled by, what decisions will be enabled by our data. So a clinician may be going to make a decision on how to treat the patient based on these data. And so you need to really understand that when you're figuring out how you're going to report your data. This is from an ICCS paper on PNH. And we talk about the reporting guidelines here. And you don't really need to read that, but basically we had a lot of discussions here on how would we report the data. And some people were saying on the paper, well, I can see that cluster and I know it's PNH. And it's like, but it's not precise. So you can't say there's this percent PNH if you're not precise. There's no meaning in that number. But they know the cluster's there and they want the clinician to understand, yeah, this is likely PNH. So that's pretty much what we decided to do here is there's a minor cluster. We cannot quantify it, but it's suggestive of PNH. So in a clinical trial, we wouldn't do that. We would just say below the limit. So these are things of, again, understanding your context of use. And so self-assessment lessons learned. This paper is also from that special issue, and that's free access. Everything's available. So the lab here decided for their CAR-T, they were going to go with 100 events, and they validated down to 0.7% positive. And now here they had a situation where they had 0.7% positive, but they only had 11 events. And so I think they probably didn't collect enough events. And so they were like, oh, now that we've gone live with the assay, well, what do we do? And here again, they had a pre-infusion and they have 0.01%, but they've got 100 events pre-infusion. So they've got some false positives here. So then you need to look at that and say, okay, what are we reporting? We validated, we didn't expect this, and that's okay. One of the big ISO things is continuous improvement. So you can change, how you're handling that. You just have to have a change control process, however that works in your institution. But this was just a nice example of, once we went live, it wasn't what we expected, despite all of our work. This is from the same lab, and they had an assay, 100 events was their criteria. It was a 1%. The only thing they changed in the assay later was the clone for the anti-CAR, which had a higher sensitivity or specificity, and they were able to go down 100-fold, like 0.001. And so again, even though they've already evaluated CAR T clones, new ones come on the market, it behooves you to also evaluate those. Okay, a couple of case studies. No. Okay, so here's some example of without applying an LLOQ, the 4 samples we did intra-assay precision, 10%, 52%, 75, and 15. So if you look at this and it's your initial validation, you're going to say, this assay is not going to meet the context of use, right? So half the samples are, we've got terrible precision. But if you then go through the trouble of validating your LLOQ and applying it, we see now, okay, these 2 samples are below the limit of quantification. When this guy needs batteries. When we apply a 0.5% LLOQ. And then it's like, okay, this assay is all right, right? The CVs are met, but we know we can't report below 0.5%. Now, in this example, we have, and this, you could think of this as instrument to instrument, analyst to analyst, or lab to lab. We have the mean of the first run and the mean of the second run. And then we take the mean of these and the percent CV. So you can see here, again, we get marginal precision here, good, and unacceptable. Now, that's without applying the LLOQ. So if we apply it, and in this case, it was 0.3%, then this sample was fine. This one had high CVs, but at the LLOQ, we can accept 33, 35% CV. Here now, both samples are below the limit of quantification, and so the CVs are not applicable, but we know both labs are reporting the same thing, below the limit of quantification. And then in this example, one lab, it was below the limit of quantification, and the other, it's really close. So we can say, okay, you know, these results are still concordant. So the assay's still performing in the other lab, but we're at or near the limit of quantification. So you can see then, as we transfer assays, how this is going to improve the success of our assay transfers. This is the assay I showed you earlier. And so for validation, we did immunomagnetic bead depletion of the CD19. We then spiked them back, and we used the FMX control to establish the LOD. And how the assay overall, we had a 1% CV. Between assays was 5%. Our limit of quantification was 0.4 cells per microliter of whole blood, and that gave us 8% CVs. And so that worked quite well. Oh dear. I don't like this pointer. I'm so used to working on my computer. Okay, I have to thank Ira for this beautiful image that he made for the cover of the book I edited, which we are doing a second version. So I know I'm going to tap out to some of you guys here in pharma to write chapters for it. Anyway, just to summarize, for rare events, we need to think of the instrument, the method, the optimization, and the validation. So it's all the things we know to do, but we really need to do them with more rigor. And now Ira made that just for me. And I was told I had to use it in all of my talks. So I'm happy to take any questions. Oh yeah, we have 1 minute.
Great. Do we have any questions here? Let me get started with, you got a lot of questions on that. So Kathy Daniels asked, for clinical samples which are sent out to CROs, what are your opinions on rare event analysis and methods for blood storage and shipping? Transfix and EDA tubes, is testing of different methods warranted, and how can we mimic rare event preservation with shipping and storage and validation stages?
Yeah, that's a very good question. And one thing is, if you're going to use, you need to titrate your antibody in your blood collection tube. So you can't titrate an EDTA and then go to transfix or struct tubes. You need to titrate in that, absolutely. And then you need to validate in that tube. And then you would do, we talk about it in CLSI, and we also have a paper on that. You would do your regular stability validation. And here's a point too, is once we establish our LLOQ, when we do stability, if a sample's below the LLOQ, we're going to just flag it as below the LLOQ, because otherwise what happens is it looks like your samples aren't stable, but it's just your measurement system is not sensitive for that level. So.
Thank you. I was wondering, well, in the examples that you showed, the samples that were below LLOQ levels, those were 2 out of 4. So, but are those studies still valid? I mean, if you've eliminated 50% of your samples, or are they just part of a bigger study where you have actually an
Yeah, that's a good question. And you can apply like what we said in the initial paper for validating methods is, you know, what you would probably do there is maybe run more samples for your validation. And again, we used to say fit for purpose, now we say context of use. So I think in a clinical trial, we thought that was okay for what we were doing. But if that's a diagnostic assay, first of all, and we talk about this in CLSI, you're going to need to run more samples to begin with. And depending on what it was for, if you're going to miss half your patients, no, it wouldn't be. It's a good question.
Thank you.
I'll do another one from online. Kathy Daniels again. For your CD4 positive T cell example, if we set tighter gates, is there a chance that we're excluding activated T cells with a different morphology? Is there any way around that?
So, I mean, on that example, it was to set the broader gate. And that's something where, you know, for your, in that development where I gave so many examples of gating, maybe they weren't good ones, but yeah, you need to, you know, figure out how you're going to gate those samples. So understand your biology of, if you think you're going to have larger or smaller cells, yes, you do need to look at that. And I did see an example once where the lab had a QC for that, and it was a regular PBMC that they were running each time. But again, the population shifted because there was downregulation of CD4, but they had their gates fixed. And it's like, okay, this isn't going to work. So yeah, just thinking of all of that ahead of time. And another thing to know is, I've done a lot of validations where I call them electronic validations, because we want to either, well, we didn't report that population or validate it, but we could have. And so we've got the data set there, or we change the gating. So you already have your samples collected. You have to write a validation plan and explain what you're doing and why, but you can go back and remind all that. And in this case, Kathy, you could be then validating with that new gating strategy. Absolutely, that would be a
great
good idea.
I'll squeeze in one last question. Is there a question? Okay, one last question then. On carryover, have you experienced or heard feedback on difference in carryover between peristaltic pump sample delivery and pressure-based sample delivery cytometers?
No, but there is like with a couple things with the carryover is, and I think I made this mistake early on, is like if you say, you know, okay, we're going to run buffer and see how many events are in the buffer. You have to be careful with that approach, because if your sample collects for, you know, TB and case, they collect in like 20 seconds. And you want to see how much carryover is, you can only do your buffer for 20 seconds because otherwise you're going to, you know, it's going to keep collecting until you reach your target value. And so that's going to look like there's more carryover than there would be, you know. So it doesn't matter what your system is. And I had a question from another webinar where it's like, yeah, if you've got an automated sample prep, you can do the carryover a couple ways. You can do it soup to nuts, or you can do each step. And it's just, again, thinking about that up front. And that's why it's good to have a team or someone else to badger the life out of you on things you hadn't thought about. So.